You can see we are in New Orleans, AHA 2016, and we're here to talk about one of the most exciting topics in cardiology, but it's probably not one that a lot of ACC members have kept up with, busy as you are. So we're here to talk about a state-of-the-art review published in the December 27th, 2016 issue of JACC, Genetics, Implications for Prevention and Management of Coronary Artery Disease. And to do so, I'm with Dr. Tim Assimes, who is an MD and a PhD, and an assistant professor of medicine at Stanford University in the Division of Cardiovascular Medicine. And you currently devote a majority of your time, don't you, to performing advanced population-based research on the genetic causes of myocardial infarction. So is that why you got involved in this?
Yes. So I've been at Stanford for about 16 years, and I think shortly after I got there, I started doing research in a field called genetic epidemiology. And so about 2003. And at that time, we still hadn't, the technology had not yet been developed to do the scanning across the entire genome, and we were still working with certain regions that we thought were interesting and often failing. So these are what we refer to the candidate genes. But we learned a lot from that. I did as well, and it was right at the beginning of my career in the field of genetic epidemiology. And then very quickly we moved into this period, very fruitful discovery period, which started about 2007, which I described in the review, where the technology was developed and we were able to then sort of in an unbiased fashion scan the entire genome looking for regions that are associating with coronary disease. And we found, I mean, depending on who you ask, if you ask the geneticists, they're happy, very happy with the number of signals, and we know that we're going to find many more in the next few years with larger and larger studies being performed. And these are proving to be very useful, first in terms of regions of the genome that we could try to use to better understand the mechanisms of disease, not only among the mechanisms that we already know about, so we're understanding more within the field of lipidology in terms of which genes should be targeted, which additional genes can be targeted other than the ones that statins and PCSK9 inhibitors target that could potentially lead to even a further reduction of coronary disease. And then that's about a third of the discoveries. And we have another 2/3 where we really have no idea, or we're starting but slowly but surely to figure that out.
Let's talk about what's already been discovered. For example, there is evidence now that common presentations of CAD are moderately heritable, especially where there's a strong family history or early onset of disease. And that really has kind of evolved from the same type of research, right?
Yes. So I think this was knowledge that was built up prior to what we call the GWAS era, the era of genome-wide scanning. People had done family studies and other types of studies that had suggested that even outside of the families that have a strong family history, the people who present with coronary disease, there's a genetic basis to that. It's not super strong, but there's obviously something there. And even if you might not see it in the family, there's definitely genes that are influencing your chance of developing coronary disease. So we knew there was something to be discovered. And now we're sort of getting a handle on how much there is to be discovered and how many genes and how many variants there are out there.
Well, another area of importance that you talk about is CAD genetic risk scores and where we are in terms of better predicting risk.
Yes, and so that part has been a little disappointing because I think going into the genome-wide scanning era, we were hoping to find perhaps a handful, 5, 10, or 15 variants across the genome that had strong effects, maybe doubling the risk, for example. But we didn't see that. In fact, the best hit, if you want to call it, increases the risk only by about 20%. And then everything else is down to around 5 to 10%. So it's in order for you to be able to improve your ability to predict who's going to get an event, you need a lot of those. You need to make a lot of those discoveries so that altogether you're able to better predict and then identify those who are most likely to benefit from treatment.
So what's got you excited in terms of where you think this is really going to help?
So I think that there's two things as I've been discussing. The first, of course, is there's a lot of new targets, new pathways that are being explored based on the information that the connections that we've made. And so I think it's great for the basic scientist who doesn't have to doubt that something in that region is problematic in terms of your risk of coronary disease. And so if an experiment doesn't work out, they'll do another one because they know that that signal is real. And I think that's extremely exciting, very reassuring, and allows for very productive basic science, which eventually should lead, hopefully, to new drugs or other types of interventions that will reduce risk. And then, of course, the second part, which will lag a bit behind, will be incorporating all these variants to just better predict who's going to get disease. And the nice thing about coronary disease is that we have a few interventions that reduce risk no matter where it's coming from, right? Healthy lifestyle, and then the use of statins. I like the analogy. The cholesterol is sort of, it's kind of when you make a cake, you need some basic ingredients. You need flour, right? So flour, the cholesterol is like the flour. So if you have high risk, lowering your cholesterol is very important in terms of reducing the chances that your plaques in your arteries will get bigger and potentially cause a problem later on. So the bottom line is, if you can predict better, then you should be able to improve overall event rates across the entire population.
I mean, you write, an exciting new era has dawned for the prevention and management of CAD utilizing genetic risk variants.
Yes.
Do you think it'll be within 5 years that we'll start seeing some clinical application to some of this, or will it be sooner, or will it be later?
I'd like to be optimistic and say within 5 or 10 years. I think all the pieces are there. It's just we have to bring them together. And so the pieces being that we need to, there's an infrastructure that needs to be developed where genetic variation can be incorporated and combined with the clinical data and then there's actually generating the genetic information itself, which used to be very expensive and now is cheap. I mean, you can get it down to about $50, which is no different than perhaps some other lab tests that we do routinely. So I think there's a lot of discussion here at the AHA, and how do we do that, and how do we move forward, and then sort of make this sort of routine genetic profiling. And there are some skeptics who think that it's still not useful, but I'm not one of them. I think there's a lot of utility there, and we ought to just take the step. We ought to stop guessing whether it's going to be useful and just start making progress towards getting that information, producing it, and then using it in a smart way.
Well, this is one of several papers that take a look at some really interesting new material, and it is the last JACC for the year. It is the December 27th issue, 2016. Please, over the holidays, take a look at the issue and follow along. For CardioSource World News and CardioSource World News Interventions, I'm Rick McGuire.